Hello class. Today's activity will be about DNA isolation from bacterial cell using boiling lysis method. First is to prepare a sterile microcentrifuge tube and label both the cap and the body of the tube. Next, we obtain fresh bacterial culture. We are extracting DNA from a broth culture in this activity. So aspirate 1 ml of the broth and transfer in the labeled microcentrifuge tube. We then centrifuge the tube for 5 minutes with its appropriate program. Always balance the centrifuge before starting. After, we then remove the tube out from the centrifuge. There should be a noticeable pellet formed at the bottom of the tube. We decant the supernatant and wash the pellet by adding 1 ml of distilled water to it. Vortex it until the pellet has completely suspended throughout the solution. After, centrifuge the tube for 5 minutes and do the washing step again. After the final washing, remove the supernatant and resuspend the pellet with 200 UL of nuclease-free water. Take the tube in a dry bath set at 100 degrees for 15 minutes. Make sure to securely seal the cap. The tube might pop open due to the buildup of pressure. After boiling, carefully take out the tube. Then, let it cool on ice. After the tube has been cooled, centrifuge it at 30 seconds. Always make sure to balance your centrifuge. Then, carefully aspirate the supernatant and transfer to a new sterile tube with the appropriate label. The supernatant is now our final extracted bacterial DNA, and use it as DNA template. We can then check the concentration and purity of this using the microplate reader and visualize the DNA using agarose gel electrophoresis. And that ends our activity. Thank you for watching.